from Microbe TV. This is Office Hours for Wednesday, July 23rd, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet. Tonight we have a special guest. I don't know if I'm allowed to say your university. Is that okay?
Yeah, it is. My
University of Saskatchewan.
That's right.
And
Perfect.
Angela Rasmussen, welcome to Office Hours.
Hi, Vincent. Thanks for having me.
I got your name up there above you. I hope I spelled it right.
You did.
Welcome, everybody. I want to welcome our moderators tonight. Les is here. Vanity Nutrition is here. So we have two so far. Thank you for coming and keeping it civilized. We appreciate it. And everyone else who's here, thanks for coming. Tell us where you are from so we can shout you out and tell everyone where everyone is from who comes to listen. Patricia's in Massachusetts. Elizabeth is in West Virginia. Kang is in Chicago. Questella, I know, is in like Iowa, Ohio, something like that. Mark Martin is here. Hello, Mark. Tacoma, Washington. Not far from where you were raised, right?
That's where I went to high school.
In Tacoma?
Yeah. It's where both of my parents are from.
Mark says it's 28C in Tacoma. It's 82F.
Beautiful.
Mike and Lori are in Seattle. There you go. John is in the Twin Cities. 26C welcomes Dr. Rasmussen. Mark says, nice to see Dr. Rasmussen, not as a tiny photo. Nope. Yeah, the little thumbnails are hard, right?
They are, and I don't like to put a picture of myself always, so I either have like an illustration or something with me wearing sunglasses.
Peter S. is in Boulder, Colorado, 25C. Jessica's at the University of Toledo. EHS Talent is in Lehigh Valley, Pennsylvania. Hypoautonomic is in Los Angeles. Les is in California. Our moderators, Les is in California. And, Vanity used to be in Long Island, but she was moving to Montana. I don't know if you're there yet, Vanity. Questella, Ohio, Indiana. Sorry, I got the next state wrong. Yeah. You see, I don't know my Midwestern geography at all. Carol, our favorite nephrologist from Bakersfield, where it is 33C. That's pretty hot. Mark Martin wants to know what high school you went to.
Bellarmine Preparatory. Go Lions.
You know that, Mark? Dean is in Wisconsin, 78F. Mark, did you go to Stadium High School? You know that one?
Yeah. I do. Stadium is actually, it's an old hotel that looks like a castle. So it was used in the movie 10 Things I Hate About You as like the main set piece.
Cheryl is from Kitchener, Ontario, 28C. MJ is from Richmond, Virginia. Rune is from Norway. Hey, there's our first faraway guest. I was going to say our first, not U.S., but Canada's not U.S., and our guest tonight is from Canada. Privyet Veronika from Moscow, 17C, with occasional super rains that have been causing floods all over the city. Yay, climate change. Logotic is in Chicago, 32C. Welcome, Angela. Gary is in Denver. Monsoon season has arrived. GSD Bio is in Encinitas, California, 22C. That's Jonathan. Airy Ozone is in Asheville, North Carolina. And Questella says, it's okay, nobody should come here. What? um, Indiana, Ohio. Really? A lot of people live there. I've been to Ohio many times.
ASV was there last year. I didn't go because it was in
Yeah,
Ohio and it was too hard to get there for me.
for you, you'd have to fly to Chicago, probably, right?
I'd have to fly to Toronto and then Chicago and then probably
Mm.
Columbus.
So you don't have any direct flights from
Just to
Saskatchewan,
Vancouver and
right?
Toronto.
Oh
Yeah.
my gosh, you're kind of
I know it's
isolated there.
terrible. I mean, I went from living in New York, where there are 3 airports, and Seattle, where there's a
Yeah.
zillion flights a day, to living in the Paris of the prairies. The airport's not as big as Charles de Gaulle.
Scott is in Bull Lake, Montana, where it's 77F. And Mark Martin says 10 things I hate about you when you
There you
mentioned
go.
your high school.
His high school. Mine is also on a hill, but it's less scenic. It's
Oh,
more
so the
Catholic.
10 things was his high school?
Yeah, his high school.
Okay. Tona is in Long Island. Welcome. Tona or Rona goes by both names. Most of these people are here every week. It's really cool.
That's awesome.
Patty's in New Hampshire on the seacoast. Markle is at the far end of Long Island where it's 22C. Renatus is in Toronto where it is 26C. Christina is in Houston, Texas. Cell and gene therapeutics viral vector. What is it? What are these keywords or something, Christina? What are you doing there? Is it a company? Is that what you work on? Anyway, welcome. And Les, our moderator, says, I did 2 live streams plus a clinical update. If you give $1 a day, that's 33 cents per program. That's cheap, right? It's a good deal. You can donate here at the live stream if you want. There's a button just below the chat with the $sign on it. You could click it and give money, or you can go to microbe.tv/contribute. Whatever you want. We'd love your support because we don't do ads. We just depend on your support. We want you. Okay. Let's see. Dion, do you know how to contact the professor? Vincent at microbe.tv. Dion is a former student of mine.
Oh, cool.
That's a good way to contact. I know I'm writing a letter for you, Dion. Okay. Don't worry about it. I'll do it.
He always waits until the last minute, Dion, I have to say from experience.
Do you know Dion?
No, but I've had you write letters for me before.
Oh, I wait to the last minute. You
Yeah,
can't
I
tell
know. You
people
wait to
that.
the last minute. No, but it's fine. I learned it from you, Vincent. The important thing is to get the letter in.
Yeah, you get it. If you get it in earlier, it doesn't help your chances, right?
No, it doesn't.
Vincent at microbe.tv. Who else here? Mark. No, not me, but my oldest son went to Stadium. Hated that movie. I grew up in Southern California, North Long Beach. Okay. Do you know Mark Martin, Angie? You
I
must,
do.
right?
I think we've met once back when I was at the University of Washington.
Luis is in Mexico City, 20C. Welcome. What's the purple crown I see? Or is it my sinus meds? I don't see a purple crown. What are you drinking there? Bourbon?
No, this is a Negroni.
Ah, Negroni. It's a good drink.
It's my favorite drink, actually. It's good all year round, and my husband made it for me when I got home.
It's Campari and what else?
Campari and red vermouth and gin.
Oh, good.
But you can make it with bourbon, and then it's called a Boulevardier.
Wow.
I hope it's okay, but it was an evening show, so I figured.
Yeah, it's okay.
Well,
it's okay. Is $18 for a salmon dinner a lot to everyone? In New York, you're going to pay $35-40.
Yeah, I mean, even in Seattle, where you can catch salmon, it's more than that usually.
Lukotic says, "How fresh is the salmon?"
Maybe you should move to Ohio and Indiana. The salmon's so cheap.
Christina, yes, clinical stage lentiviral gene therapeutics. Okay, so Christina's working for a company doing that. Thank you very much.
Cool.
Oh yes. So on the chat, they now have crowns for number 1, number 2, and number 3 fans. If you make a lot of comments, you get 1, 2, and 3. That's what Chris, who was saying it? Patricia was talking about the purple crown. Yeah, it's weird. I just noticed it today. Something new. Elena, privyet. There's the Cyrillic privyet from Reno, Nevada, 29C. I've been to Reno. It's hot. Les is stumped. No, the crown is number 1, number 2, and number 3 fans. If you make a lot of comments, basically, you get elevated to that status. MN is in DC, where it's a nice evening outside. Mark says, "Oh my God, I hope I was okay when you met me."
You were wearing a fun necktie. I remember that. And you were talking about some science fiction authors that I know. So I don't remember anything bad.
Mark, you're always okay. Don't worry about it. Tona says, on Long Island, a salmon dinner is at least $25. Yep. What's a salmon dinner in Saskatchewan?
I don't really eat salmon here. I'm
Because
from the
it's
Pacific.
too far
Well,
inland?
yeah, it's too far inland. They actually do have some steelhead farms here, so steelhead
Yeah.
trout.
Hmm.
And we go fishing for lake trout. So I'll eat that. But if we get salmon, we usually make it ourselves because we order it like from BC. And I'm just really picky about my salmon. I was spoiled growing up in the Pacific Northwest.
Seattle, right?
Yeah, I mean, I'm really more from Tacoma, but Tacoma also is on the Puget Sound. And every year, this was way before Twilight, so I thought it usually sucked, but we would go salmon
Hmm.
fishing in the summers off of La Push, which
Yeah.
is up on the Olympic Peninsula, and did catch some amazing salmon.
Okay, Dion, don't worry. I'll get it in. It's due next week. I'll do it on the weekend, okay? No
Trust me,
problem.
Dion, he'll do it. Like it just you'll wait, till the last minute, like I do now when people ask me for letters, but it always gets done.
I have a good track record of my students, at least in the virology course, getting into medical schools. Probably has
That's
nothing to
awesome.
do with me, but if you do well in the course, I say that, and that might help. Mark says, excellent choice for a cocktail. Okay, Vanity says the crowns display cumulative points you've earned for chat engagement. So another ploy by YouTube to get more chat engagement. That's a good thing, right? Jeff is in Elgin, Illinois, where it's 33C. Stylin' with the Negroni.
I'm gonna have to
Hans
make
is
another one if I keep like drinking every time somebody comments on the Negroni.
Hans is in Ecuador, where it is 28C. Welcome, Hans. Jefferson Space is in Miami, where it is very warm. Questel, I got spoiled by a small town place that recently closed. Meals were only like $8 to $12.
Gotta move there.
Well, maybe there's nothing to do. That's the problem, right?
Yeah.
Scott
Although I
says
live in a place right
Costco.
now with kind of nothing to do.
Really?
Well, compared to New York City, I mean, I guess,
Mm,
it's
yeah.
a tough comparison to make.
Scott says Costco deli, 69 cents an ounce. An ounce. Wow. And you have to cook it, though. Joel, thank you for your support of science communication. Come on, people. Time to support the show. Appreciate it. John, thank you for your support of science communication. I've became a fan of the good doctor listening to her on NPR. That'd be you.
Yeah, I know.
And
I was
reading
like, it's talking about me.
and reading expert information online about pandemic origins.
I'm
We'll
glad to
talk
help John.
we'll talk a little bit about pandemic origins today. Scott, thank you for your support of science communications. Sunglasses perpetually fall onto video game controllers. Proud face. I think that's describing the little emoji. Yeah. Yeah, it's sunglasses falling onto a video game
Oh yeah,
controller's face.
I know exactly the one.
Dean, hello from Oak Park, Illinois, 34C. Airy Ozone thinks there's a character in Twilight named Angela. I don't know what Twilight is. Do you?
Oh, it's that book about like the vampires that sparkle and the
Oh,
indigenous werewolves. It takes place in a town called Forks, Washington, which is a town that I also didn't like very much when we would travel through there on our way to La Push to go fishing. But
yeah.
basically, like this girl moves to Forks. There's vampires at the school. She falls in love with one of them. He's really creepy and stalks her. And I didn't read any of the other books. And then she has like sort of a love triangle going with like the indigenous werewolf who lives over at La Push, which in real life is a Quileute reserve. So it's the Quileute tribe is from there. And so the Twilight author, who is not from Washington State, decided to make them into werewolves. And then the random vampires moved to Forks. Actually, one of them became a vampire, though. I do remember this because he was about to die from 1918. And so, like, his girlfriend made a vampire.
Christina's in Houston where it's 100 degrees F. Okay, Hans has got a question, but we're gonna remember this and get it later because there are a few other things. What's the single most clear proof that SARS-CoV-2 did not come from a lab? Can you say it in like one sentence?
No, but there is no proof that it didn't come from a lab. There's a collection of evidence that's not consistent with it coming from a lab.
Okay. Vanity, I'm an example of med school acceptee who you wrote a letter for. That's right, I did. Now, so Vanity took my course online. I think it was during the pandemic, right? But then she became a moderator for this, showing good knowledge. That kind of thing is great to put in a letter, right? Do something beyond the course. So that's easy to write. Noir, welcome. Nice to see you again. Joel is in Bath, UK. Welcome,
It's
Joel.
late for you.
It's late for the Europeans, and we usually have someone from Norway. I think we did have someone from. It's like 2 a.m. in Norway. Yeah.
Rune.
It's 24 degrees. SRR is in Paso Robles. Great wine. Did you ever have Paso Robles wine, Angie?
I have. My older stepson went to Cal Poly, so when we go visit him, Alexi and I would take a little field trip to Paso Robles. Is it Robles or Robles? People there pronounce it Robles, but I think in Spanish it should be pronounced Robles.
Robles.
Anyway, I've been there. They have some great wineries and some great wine.
Griffin says that Twilight is a work of art.
I mean, I think Beverly Hills 90210 is a work of art. So there's, no judgment here. Just wasn't my cup of tea.
Elena says, I used to live and do field work on the east coast of Siberia. Speaking of salmon snobs, I'm right here with you. Fishing there in mid-1980s was fantastic.
I bet. I have always wanted to go there. My husband is from Russia, and I asked him if he'd ever been to the Kamchatka Peninsula, and he laughed in my face and said no, because that's like a big military installation. But they also film Wild Russia there. And there are amazing salmon being pulled out of these streams by these giant Russian bears.
Can you go to Russia now? Most Americans cannot.
No.
They'll send you back. Yeah.
No Russia anytime soon, sadly. But I've always wanted to go see that, just because I know how beautiful the Pacific Northwest is, and it looks like similar but different. So I'd like to see it someday.
So your Twilight, according to Lagodic.
I mean, I read the book, and it was like a pop culture thing when I was, younger, I guess. I was still in my 20s, I think, but I did read it, and I did not really enjoy it, mostly because I also thought it was a ridiculous depiction of Forks and La Push, Washington.
SRR, thank you for your support of science communication. Really appreciate it. So hypoautonomic says they typically pronounce it the non-Spanish way. Okay. Okay. Margot is in Phoenix Metro, 103. You got the temperature win today, Margot. Tona wants some background on you. We'll get to that in a second. And Vanity was in my spring 21 class. I remember that. Did you see that not so great video 8 days ago, unmasking COVID-19's true origin? Did you see
Yeah,
that?
we were both in it. Yeah, so I did.
Oh, it's not so great according to EHS Talent. Oh my gosh. That's
I
the
mean,
one we are in.
I think we looked good. Like
So I
a
didn't watch
journalist
it yet.
friend of mine said stunning women. So I was like, good, I think that includes me. So at least I looked good.
The problem is EHS Talent that. So when I was approached by the producer, he said, we want to have 50-50 lab leak and 50-50, half lab leak and half natural origin. I said, no, if you do that, you're not going to get any natural origin people. So he said, okay, and they didn't. But then after they filmed me, then they started getting some lab leak people in. And I don't think that adds anything to it. It would have been cool if it were just a pure factual documentary, right? Oh, well, did you watch it, EHS Talent? So did they film you in Montreux?
No, they didn't
You
actually. That
weren't?
was
Were you
in,
there?
I was in
Okay.
Montreux. In fact, in Montreux, Vincent, is where you talked me into doing it because I wasn't going to do it.
Yeah.
And then you said, well, what if like I'm the only one, me and Mike Warby are the only ones who are speaking actually for the science. And
Yeah.
the rest of it is, all these lab leak people. And so I said, okay, I'll do it. And then they cut Mike Warby out of it. They added additional lab leak people and including some of the crazier lab leak people.
Mm.
And they weren't really very critical about talking about people's backgrounds. They introduced Emily Kopp and Paul Thacker as journalists without mentioning that Emily Kopp is a journalist previously for an anti-GMO activist organization and currently for the Daily Caller. And Paul Thacker is basically a right-wing mouthpiece who has written, I'm surprised he hasn't written a hit piece about you, but he's written one about me, about Christian Anderson, about Amy Maxman, about basically anybody who crosses his path that he doesn't agree with.
But Angela, you know that Ebright said I'm irrelevant now, so nobody's going to write about me.
You're
What is he?
irrelevant. We're together, oak and acorn, tree and nut. What he said.
Oh, Visto is from Sydney. Welcome. Sydney, Australia. Welcome. 5 degrees C. It's winter down there. Yep. So SRR says in Paso, we pronounce it like Dr. Rasmussen said, ignore the tourists. Okay, so it's Robles. Paso Robles. Okay.
It was weird for me, but my stepson kept pronouncing it that way, so I wanted to fit in.
Luis is in Guatemala, 25C. Any comments on the S2M deletion during the rise of Omicron? I don't have any comments. Do you?
No,
Nope.
I mean, these deletions happen. Omicron's super different. And now, like, talking about Omicron, like, how many viruses are we actually talking about here? Probably a lot.
Why do you think COVID is not a vascular disease? So I say, that's what I say. There are studies that show it is. Risk of heart attack and stroke go up for months after infection. One of my doctors called it vascular. Well, just because your doctor called it vascular, I'm not trying to insult you, Melinda, doesn't mean that it's vascular. It does have cardiovascular effects. But when people say vascular, they say it replicates in the endothelium throughout the body. And I just don't buy that. I just don't see evidence for that. Do you, Angie?
Not really. I mean, it does, I'm sure it does replicate in the vascular endothelium in some places, but I actually think about it like Ebola, even though obviously COVID is not like Ebola virus disease. In Ebola, the mechanism for pathogenesis of the hemorrhagic disease is actually excessive systemic inflammation. That causes the blood to start clotting indiscriminately. It causes the blood vessels to start leaking. And they've actually done studies where they explanted like vasculature, like blood vessels from non-human primates, and infected them with Ebola, and showed that essentially the vascular leakage and the hemorrhagic disease is not caused by the direct death of those endothelial cells.
Yeah.
So, I mean, I think that, like you said, it has vascular effects. I mean, it's a virus that infects different tissues and has different tropism. But when you're thinking about the disease that it causes, you have to think about the whole organism. And there can be disease in tissues that actually aren't directly infected.
Yeah.
And I think, COVID it's just a very good virus at infecting a lot of different species and a lot of different tissues and causing a lot of different responses in a lot of different hosts. So it's
Well,
not a I wouldn't call it a vascular disease either.
there are neurological symptoms, but it does not replicate in the brain. Those are probably due to hypoxia and inflammation, right?
Right.
But you can barely find infectious virus particles in the brain.
Yeah, and it's so hard to tell in some of these studies because when you're working with a brain, obviously, unless you have a biopsy, I mean, most of the time you're working with autopsy tissues, and the brain's pretty big. Like, so unless you get like exactly the right spot where a virus might be replicating, it's not necessarily going to be disseminated throughout the
Right.
entire organ, and you're not necessarily going to see a lot of it. So I wouldn't exclude the possibility that there's persistent infection in the brain, but I wouldn't necessarily say that there's really conclusive proof of that. I mean, the group of people that claim that there's persistent infection, I'm just not totally convinced by a lot of that data.
I mean, it's the same for the GI tract. A lot of people think there's persistent infection, but it's very little infectious virus there. So until you do that, it's not proven, right?
Yeah, and I mean, shedding RNA is not the same thing as shedding an infectious virus.
Oh,
Another
yeah. I
another
mean, that's the
lesson I
big
learned
problem.
from you. You
That's the
always
big
got to do the
problem.
plaque assay.
Yeah. Tona says, what podcast was Vincent just on? Well, that was the Mark Thompson. I just queued up a link. So he does a long live stream and people appear at different times. So I put a link in with the timestamp. So if you go there, you can start playing. Yeah, that was this week. Christina says, I'm interested if anyone has read Ulrich Kammerer's research in Science Public Health Policy and the Law on SV40 promoter enhancer sequences in RNA-based COVID-19 lots. Do you know anything about this?
I don't know about Ulrich Kammerer's research. I do know about some of the SV40 stuff, and it's not particularly convincing. I'll put it that way. And it doesn't cause turbo cancer. I don't see actually why. I guess it depends on which vaccine we're talking about, but I'm assuming this is mRNA vaccines. I don't know why SV40 promoter enhancer sequences would be in an mRNA vaccine because they're synthesized. They're not coming from a plasmid, which is the only way that you would really get SV40 sequences in there unless you were working with a non-human primate cell line that was infected with it. And they also don't require a cell to make. So, I think a lot of that it's kind of stuff that like people like Robert Malone have made up to make these claims of, these vaccines are incredibly dangerous when they're actually not.
Well, thank you, Noir, for saying that I will never be irrelevant. I appreciate it. Okay, Mark says, in Southern California, listen to how people pronounce Tijuana or La Jolla. What do they say, Tijuana or La Jolla? Okay. Luis, I finishing my thesis. Will soon take my professional master exam. Your classes on flavies help me a lot. I'll thank you in my thesis. Thank you so much. Yeah, microclots in long COVID is a problem. Yeah. They can have a lot of different origins.
Yeah, that's one of the reasons it reminds me of Ebola, actually, because there's in Ebola, like it's through the whole body, but there is disseminated intravascular coagulation that that basically causes microclots everywhere, and it eventually makes it so that you end up in shock and die in many cases. But that's kind of what makes me make that connection between COVID and Ebola.
All right, I'm going to pause on the questions here because you have to tell us a little bit about your background. We know you grew up in the Seattle area, right?
Yep, I was actually born in Tacoma as well, but I'm from a town called Puyallup, Washington, which is about 15 minutes from Tacoma.
And you went to college, I think, out east, right?
I did. So that was the first time I ever moved like to the East Coast, and I went to Smith College. And I can't believe how long it's been, but I attended my 25th year reunion this May, which is weird because I don't feel 25 years older necessarily. But it was a great experience. I met some of my best friends there, but also I worked with my advisor, Christine White Ziegler, who really taught me a lot of fundamentals about microbiology. She studies transcriptional regulation of uropathogenic E. coli fimbrial gene operons, or at least that's what I worked on. So I got some really basic cloning education, some basic molecular biology experience. I learned a lot of really classical microbiology techniques like using phage to move things around and doing chemical-based mutagenesis and replica plating, doing a lot of Miller assays, the good old LAC-Z operon and that reporter system. So it was a great experience, but I didn't want to work on bacteria. So then I moved back home after college, and I got my own apartment actually in Tacoma. And I worked at a small biotech startup in Seattle that was developing at the time polyclonal autologous T-cell therapy. The company went under. There's better T-cell therapies out there for cancer now, but I did get a solid background in flow cytometry and immunology while I was there. The technology lives on. Anti-CD3 and anti-CD28 Dynabeads, you can still buy them from Thermo Fisher. And amplify as many T cells as you want from PBMCs or a leukapheresis product with them. Then I decided there was a riff at the company between different rounds of funding, and I realized also at the same time that I wasn't going to be able to sort of break through, like to be able to do work that I wanted to do if I didn't go get a PhD. So I thought that was probably a good time to start applying to graduate school. A bunch of my friends from Smith lived in New York, and I was like, oh, it would be really fun to live in New York and also like get paid to go to school. That was a pretty naive way of looking at graduate school, as I learned, but it was a great experience going to graduate school in New York City. And of course, I was in Vincent's lab at Columbia from 2003 to 2009. When I finally, after a very difficult project, managed to clone a virus that was mouse adapted. It was human rhinovirus 1A, and started to passage it through mice, but then I graduated. Then I went to the University of Washington, so back home again. But this time I lived in Seattle. I was in Michael Cates's lab in the Department of Microbiology, and I worked for my postdoc on hepatitis C. And the Cates lab was a systems biology lab doing, at the time, microarrays to look at global gene expression in response to infection. And that originally was what my graduate school project was supposed to be, if you recall, Vincent, because we had a mouse-adapted rhinovirus, rhinovirus 16L, and we had a transgenic mouse that expressed the receptor for that mouse-adapted rhinovirus, but it unfortunately didn't express the transgene. So I thought I was going to come in, like get a paper real fast, like make a mouse model of pathogenesis for the common cold, and then start doing Affymetrix gene chip microarrays on it. And I was going to be getting Nature papers all over the place. And, science just doesn't work out that way sometimes. But I stayed at the University of Washington after I was done with my postdoc. I was there for 3 years as a postdoc, working on hepatitis C virus. Towards the end of my postdoc, they developed new classes of drugs that actually worked to completely get rid of hepatitis C. And as a result, there wasn't as much funding for hepatitis C virus anymore. So that's when I switched to working on emerging viruses. And at first I was a staff scientist who managed at the time it was called the Pacific Northwest Regional Center of Excellence the RCE program. The idea was that NIAID was going to fund all these centers in different parts of the country, and we were going to like respond if there was, a biotech incident in the Pacific Northwest, I guess. Like if somebody needed some research or some microarrays done, I guess we would be standing by, ready to do it. But we did some cool work, and that's when I started working on emerging viruses and started working on Ebola, and I did a little bit of work on dengue, did a little bit of work on flu, and also MERS coronavirus. So and some others, Lassa, Lujo, like random hemorrhagic fever viruses here and
Mm.
there. But that was really when I got into the host response. And then I got my first faculty position at the University of Washington, which was research track. And then I moved back to Columbia and the Columbia Mailman School of Public Health and the Center for Infection and Immunity that is run by Ian Lipkin. And I continued doing that work there. And then during the pandemic, I had to make a decision because when I lived in New York, my husband and my family still lived in Seattle. So I was commuting back and forth. And it was kind of a no-brainer. Like, I didn't know if I would be able to even get into the lab. The BSL-3 lab there at Columbia isn't very large. So I decided to stick it out with my family instead of staying in New York and possibly being stuck in my apartment by myself. And I didn't really have that much to do besides like read preprints and talk to the media. So that's how I kind of ended up becoming, I guess, famous, like Z-list famous on Twitter. But that's why I was on the news a lot in 2020. And it was really great. It raised my profile in many ways, even though it was like one of the most stressful years of my life. And ended up getting recruited to Vito. And my younger stepson graduated from high school, so my husband didn't need to stay in Seattle anymore. Also, his work went fully remote. So he's from Russia, like I said, so he didn't mind the fact that it's minus 40 degrees here in the winter and that there's lots of hockey in Canada. So we moved here in April of 2021, and now I'm a principal scientist at the Vaccine and Infectious Disease Organization at the University of Saskatchewan, where I'm also a professor in the Department of Biochemistry, Microbiology, and Immunology. So I have students at the Vaccine Research Institute I work at, and the reason I moved here is because we operate one of North America's largest containment labs, and we're in the process of upgrading to maximum containment. So I'll be able to actually do my own Ebola work rather than having to rely on collaborators. So that's kind of it in a nutshell, and I'm happy to answer any questions.
What do you work on in your lab?
So right now, we have projects going on still on SARS-2, and we're working on a question that I've been interested in back since I was working on Ebola. I started working on Ebola in about 2013. Published in 2014 a model called the collaborative cross that's a panel of genetically different mice. And we found that basically when you give Ebola, the same dose of Ebola by the same route to these mice with different genetic backgrounds will get different symptoms, basically. Some of them will just die. Some of them will die with hemorrhagic disease that looks a lot like most severe cases of human Ebola virus disease. And some of them actually just like lose a little bit of weight and then they get better. It's like the same phenotype that you would expect from, I don't know, infecting them with like a mild strain of seasonal flu. So I observed in some of our Ebola experiments that there were differences in outcome between male and female mice. And we've actually expanded that into a much larger research program since after the pandemic, it became a lot harder to do BSL-4 work in collaboration with other people because those facilities were, during the pandemic, like everybody else, devoted to working on SARS-2. But then afterwards, my longtime collaborator, Heinz Feldman, stopped working on Ebola virus and is now working on other viruses, so Crimean Congo hemorrhagic fever virus and things like that. So I figured I better work on some BSL-3 viruses and start looking at this sex bias and seeing if that's a thing too. My student actually just gave a flash talk about it at ASV. She just published a preprint on sex bias in MERS using the HDPP4 mouse model. We have more studies coming down the pike in vitro that are looking at the effect of sex hormones on SARS-1, SARS-2, and MERS coronavirus infection. hopefully we'll be able to expand that also to H5 and some of the other viruses that I'm working with. We've had a very long ordeal because I was unable to get my security clearance renewed for 2 years because there is a huge backlog in Canada. And I've written papers with people who also have appointments in China. And my husband is from Russia. So I had to undergo extra scrutiny. And for that reason, I have been severely delayed in getting started with an Mpox project that I have, and that is to look at a lot of that stuff in Mpox and actually to use the collaborative cross to develop a model of Mpox, because Bernie Moss at NIH has developed a model of Mpox in the cast mouse, and the cast mouse contributed some of its genetics to the collaborative cross. So we're going to get started on that now, finally, that I do have my security clearance renewed. So we're doing that. In the meantime, though, we started working on some in vitro projects with Mpox, looking at the effect of ApoBec editing on the viral genome, and then in turn the effect on host responses. Because Andrew Rambo and Anya O'Toole, postdoc in his lab, had analyzed sequences from the clade 2B epidemic and saw that they had way more mutations than you would expect from a DNA virus like that. And they were all these mutations that were indicative of ApoBec3. But they don't seem to have that in like some of the cases that are known to be from zoonotic spillover. And Andrew has hypothesized that basically rodents, which are the normal reservoir for Mpox, have one gene for ApoBec3, whereas humans have 7. So we're actually overexpressing ApoBec3 in lentiviruses, and testing this now while also using RNA sequencing to look at the host responses and how some of these different ApoBecs might actually affect not just the virus and its evolution, but also how the host is responding to it.
Yeah,
and then we've got some preliminary.
I think we did that paper on TWIV, actually. So the idea is that ApoBec mutations are a signature of human circulation, right?
Yes, exactly. So if it just spills over from the rodents, then you would expect to see, this much slower rate of mutation. And you would expect to see mutations that are not characteristic of the cytidine deaminase activity that ApoBec has.
Got it. Okay, let's see. We have lots of questions here. Tona wants to know what's going on with the 25 cases of rabies in Nassau County. This is very interesting because it was previously gone, of course, in people. And there are both people and raccoons, I think, that have come down with rabies. I don't know why suddenly we have 25 cases. No idea. I wonder if Daniel would have any insight. Tona, send Daniel an email. Daniel at microbe.tv. He'd probably love to talk about that since he lives on Long Island.
Yeah, I mean, if there's a bunch of rabid dogs running around there, like I'd be careful.
Yeah, it's a problem. Melinda says, did you guys see the study about virus at the brain edge and schizophrenia? Have you heard about it? So in some patients with schizophrenia, in the outer membrane of the brain, they find hepatitis C virus.
Really?
I don't know if it's weird. Has anything to do with it, or if it's causal or associated. Who knows? But it's intriguing. Same thing with
specific for hepatocytes.
I know. So it's probably not replicating there. It just got there somehow, right?
I mean, it does circulate in the blood.
You're absolutely right. Because hepatocytes have that microRNA, miR-122.
Yeah, 122.
And other cells do not, and so it won't replicate elsewhere.
And that virus is such a pain to grow. Like, really. Like, I'm not sure. I should have asked Charlie when I saw him at ASV, but I'm pretty sure that JFH1 is still the main virus that anybody actually knows how to grow.
That's right.
Because of miR-122, but it's not like with the rhinoviruses where I could just pick one, like, and have a different virus that I could work with. Like there's JFH1 and that's it pretty much.
Which is not good because there can obviously be differences, right?
Yeah.
Intisar says, from San Diego, how much immunology do you know from learning virology? How do you keep everything straight in your mind regarding the tons of viruses you read about? You could take that, Angie.
So I know a little more about immunology, not just from virology, but because I had this job as an immunology technician, basically at an immunology company. So I still remember some of the stuff about T cells. And I used to actually think that I wanted to be an immunologist. And then I got to Columbia and I started seeing the immunology talks, which, again, nothing wrong with it. Really important work, but not my cup of tea to look at every developmental stage with flow cytometry from, the bone marrow into something becoming like a very specific type of effector cell.
Mm.
So there's, at Columbia, there's a lot of developmental immunology. And again, I'm more interested in immunology when it's like, hey, we're getting rid of this virus. So I certainly know a lot about immunology, and I especially know about the host responses that occur after a viral infection, because we're not restricting host response studies in my lab to only immune responses, but a lot of the responses, obviously, that occur after an infection are immune responses. So I've picked up quite a lot of immunology, but I would not call myself a hardcore immunologist just because immunology is extremely complicated and people are really, really specialized and know more
Mm.
than I've ever learned. But that's also how I kind of keep everything straight in my mind regarding the tons of viruses that I read about and work on. I forget a lot, and then I just have to look it back up.
Yeah, if you don't use it, you're going to forget it. But
Yeah.
everything I teach every year, I remember because I talk about it every year. And even if I do a paper on a podcast, I'm not going to remember it unless I perpetuate it, right? But immunology is interesting. When I was younger, most of virology was molecular virology, understanding how viruses replicate in cells. And now it's much less of that and more pathogenesis. And to do pathogenesis, you need to know immunology. It's an essential part of it. So a lot of the papers we do are viral pathogenesis. So there's a huge immune component. And we end up doing lots of immunology on the podcast, right?
Well, I'd say that I'd make the argument that, and some virologists would probably disagree with me, but because I look at it from the perspective of the host, I'd argue that almost every single viral disease, like the pathogenesis of every virus, is actually driven by the host and
Yeah. Yeah.
oftentimes by the dysregulation of host immune or inflammatory responses.
John wants to know what's the best time to get the flu shot this fall?
As soon as you can,
Of course, if you're
prevent
older,
reassortment.
if you're older, though, your immunity is going to
Wayne.
subside. And so, older people end up getting it in August, right? Because they're told you're old, you have to go get a flu shot. But then it wanes by December, January, and then that's still flu season. So Daniel says some people get 2 shots in the winter because of that. But if you're
Really?
young,
Terrible
John,
idea.
no, John, you could get it whenever you want. Whenever it's not available yet. It's just July, but it should be available in August, right?
Just enjoy it while it lasts, because you might need to come to Canada next year to get one.
Oh gosh, going to be the flights to Canada are going to be really expensive.
It's going to be horrible. Maybe Air Canada will actually make some more flights, though.
Mark, welcome from San Jose, California. I don't know if you remember the early days of TWIV, Angie. We used to have Drobo as a sponsor. Those hard drive arrays, and Mark used to work for Drobo, and he gave us a
Oh,
couple to give
really?
away. Yep. Okay,
I
here's
remember.
one for you, Angie. Is turbo cancer a proven form of cancer?
No. This is just, it's made up. I'm not sure if I can swear here, but it's made up crap. It's
Crap is
just,
okay. Yeah.
yeah, it's just something that, people came up with to justify, I guess, this also made up contention that people are like dying suddenly after getting a vaccine, or in fact, there's a movie called Died Suddenly. Turbo cancer is like a slightly longer version of Died Suddenly, but it's basically the idea that like you get cancer from a vaccine almost immediately, and then like within a month you're gone. And that has not happened as far as I'm aware. And probably anybody who got cancer around the time that they got their vaccine and had a very rapidly progressing disease did not get that cancer because of the vaccine.
Right. It's just so
Certainly,
much,
SV40 does not cause turbo cancer. Sometimes it's not even very good at immortalizing cells.
so much gaslighting in COVID. It's just unbelievable. It started with the Great Barrington Report, that baloney document with no science behind it, and it's been continuing ever since, right?
It's not even a report, it's a declaration.
It's a
Um,
declaration, right?
so self-important and stupid. And like, if you read it, I mean, it doesn't, it's not specific about anything. It's like, oh, lock up some old people or vulnerable people, which is never defined. And then everybody else enjoy the benefits of herd immunity after you all die of COVID. Terrible.
So there was a counter document. Do you remember the Jon Snow
I signed it.
response? And one of the co-authors wrote into Daniel's program today because we were talking about how she got no attention. That document got no attention, yet every claim made in the document was backed up with scientific references, in contrast to this Great Barrington Declaration, which had no science. It was just stupid ideas by people who didn't know anything about viruses.
Well, you know, we just published the Jon Snow memo in the Lancet. We didn't have like a libertarian economic think tank like underwriting the Jon Snow memo. So
right.
I wasn't one of the authors who originally drafted it, but I was like in the appendix of signatories. And I mean, it was just people who were like, we were all mad about the Great Barrington Declaration because it was such a dangerous idea. Somebody, Kit Yates from the UK, has done modeling that basically has showed that in the US, if they had implemented the Great Barrington Declaration, there would have been 1 million additional deaths. And I think, you know, we didn't have the numbers to put behind it then, but everybody was just really horrified by it. And that was the motivation for writing it, not the fact that this libertarian think tank decided that they wanted to reopen the economy and funded us to do it.
Here we have a bogus document with no scientific basis. Bhattacharya is the author. Now he's head of NIH. And Kuldorf, another author, is on ACIP. And wasn't Prasad on that document also?
No, it was Sunitra Gupta, who is, in fairness, like not really doing anything, but she's in the UK. She's at Oxford University, so she couldn't really be in the US. But, you know, I mean, the three of them, and none of them actually have any relevant experience. Sunitra Gupta has the most because she is a theoretical epidemiologist and she's modeled outbreaks before, but had never really modeled, you know, she models other diseases too, like diabetes and things like that, obesity. So she's not exclusively infectious disease. None of them had any virology expertise. Martin Kuldorf is basically a mathematician, and Bhattacharya is a doctor who's never seen a patient, who also is more or less an economist.
He's an economist. He wanted to get the country opened again. That's all he was interested in.
Well, and it wasn't the first time he took money to do it either, because he took money from the CEO of JetBlue to do the Santa Clara study, which was a really poorly done and poorly controlled serology study that they claimed showed that way more people had COVID. And so it must not be that bad. So let's reopen everything.
It's an example of a document written in the absence of information. We didn't know anything about long COVID. We didn't know anything about disease in younger people, and they assumed none of that happened. And so it's really idiotic. But anyway.
There were so many things like that were actually provided by economists. I mean, Emily Oster, who's an economist at Brown, wrote in the summer of 2020. And I mean, I remember reading this and I was just like, you've got to be kidding me, that children do not get infected with COVID or SARS-2. And it's like, you have to understand there is a difference between getting sick from a virus and getting infected with it. And just because you don't get sick doesn't mean you can't transmit it to somebody. In fact, that's the entire reason that SARS-2 became a pandemic, in my opinion, because you can transmit it before you know that you have it.
Yeah, exactly.
And you know, to think that little kids, basically, who are famous for spreading respiratory viruses, would somehow not give COVID to grandma and grandpa was absolutely absurd to me.
Okay, here's a non-viral question. Isn't filarial worm reproduction also called shedding? Yeah, when the worms shed the outer cuticle, it's part of reproduction. But the reproduction is more than just shedding.
I'm glad you knew the answer to that one, Vincent, because I do
Well, I
not.
do a podcast called This Week in Parasitism. I do
Right.
absorb something. And my old buddy Dixon taught me a lot.
Yeah, I
You know,
miss Vic.
Dixon passed away, right?
I do. That was really, really sad. He was always such a nice man. He was always so kind to me when I was a graduate student, and I was like really scared of him because he was so intimidating. I know I don't seem like I get scared of people, but I do actually get scared of people who are just have really great reputations, or I used to. These days I don't. But when I was a graduate student, I was always, I don't know. I mean, I worked for you, Vincent, so I was used to being around you. But Dixon came in and he had all this vertical farming stuff, and he'd like done a TED talk, and that was like right when TED talks were like
Mm,
becoming like the cool thing. And he just always seemed so distinguished and professorial, and he was so kind. He was just such a nice man. So
he
I was
was.
really sad to see that.
He was a nice man. So Tony says, I'm on the Mark Thompson podcast in an hour and 2 minutes. They spent like 5 minutes figuring out how to pronounce my name.
Racaniello,
Yeah, but neither he nor his producer could get it right.
did he
That's
do it
okay.
like extra Italian-y, like
No,
Racaniello?
no. I mean, first they were doing Racanello, which without the "ny," and then the first part rock. They were calling me Rockanello. So none of the two. Here's a question for you, Angie, since you were so well trained. How are cell lines selected for plaque assays? Is it a trial and error, or is there a systematic approach?
Well, I guess it depends. Oh, so novel virus, right? So if it's polio, like you just use HeLAs like everybody has for years. But if it's a novel virus, well, then I mean, I guess there's a systematic approach, but it's also at least the approach I would take would be like grab the cells that you happen to have in your lab
Yeah,
and try
that's
it.
right. That's right.
um,
That's right.
unless I had some reason to think like this infects this type of cell or, it infects this thing, then I'd probably find the one that was the easiest to use. But because I've had to develop plaque assays from not on a novel virus, but from a virus that we weren't used to plaquing in the lab and people hadn't really worked with that much, the rhinovirus 1A, it was a lot of trial and error. I knew the cell type that would work because I knew that it
Mm,
infected HeLa cells and caused cytopathic effect, but it was really hard to get it to get just the right conditions to plaque it. We had to buy, I don't know if you remember this, Vincent, but we had to buy this stuff called type 7 agarose
oh my
that was like
gosh.
unbelievably expensive, and it was the
Yes.
only agarose that it would plaque with. Like
Yep. I
we
remember.
tried all this other stuff. Like we were using different kinds of agarose. We were borrowing from other people's labs who were doing this, like they need super clean agarose to do their like special molecular biology with, and we were borrowing that and trying it, and nothing worked except for this type 7 agarose, and it was just a lot of banging my head into the wall and doing a lot of plaque assays over and over again until I got one that worked. But if you sometimes you can't get one to work. When I was a postdoc, I had this plaque assay protocol that I got from Juliet, who Juliet Morrison is an assistant professor at UC Riverside and was also a graduate student in Vincent's lab when I was there, and was one of my close friends. Juliet had given me this protocol for plaquing dengue on BHK21 cells, which are baby hamster kidney cells. And I just could not get that plaque assay to work consistently. And it probably was due to the fact that her BHK21 cells in Adolfo Garcia Sastra's lab at the time were different from the
Mm,
BHK21 cells that I had at the University of Washington. And our techniques might have been slightly different, whatever. I was actually using a different virus isolate than she was, so that could have been it too. But I just said, I'm so tired of trying to develop more plaque assays and screwing around with these plaque assays. So I developed a focus forming assay, which is like a plaque assay, except instead of looking for cytopathic effect, you stain it with antibodies, and then you can put on like a immunohistochemical substrate that turns brown or whatever, purple in my case, on these plates. And so you count in this case, you counted the purple spots instead of counting the clear spots against the purple background. But you can also do something called a median tissue culture infectious dose assay, which is sort of the standard practice at the institution I'm at now. But I've realized that I don't like doing TCID 50s because I don't think they're precise enough. So I'm training my postdoc to work in containment. And together we're going to optimize a plaque assay for SARS-2 because I'm old school and I love me some plaque assays.
Tells you a lot of information.
It does. And I mean, the TCID 50s are okay. They definitely give you a measure of infectious virus.
Yeah.
But I don't like this whole converting TCID 50 units to plaque units. And I'd rather just count some plaques. We do it for Mpox. And oh, it just felt like old times, like counting plaques.
Carol wants to know if there is any evidence that we will have a bird flu pandemic.
I mean, there's a lot of evidence that we could, but I tell this to every reporter I talk to on this topic, and I told it to 2 reporters today, that we could have one tomorrow and we could never have one. We just don't know. The viruses that are circulating now can definitely infect humans. They're definitely more capable of infecting mammals, the ones in North America for sure. There have been a rash of cases in Cambodia and Southeast Asia. It's a different clade of H5N1, but it's a very similar virus. It just has a different HA segment on it. Here, there are viruses. We have not seen a slowdown in the number of wild animals that are being found dead in North America. The birds still definitely have it. I don't know if people are getting it because the CDC is not testing anymore. And they are testing in Canada, but nobody's gotten it. Although we don't have it in dairy cows up here. And right now it's not flu season for birds either. The birds have it. But the real problem with the wild birds is that a lot of them can get it and they don't get very sick from it. So ducks in particular are the big bad wild birds when it comes to H5N1 transmission because they're social, they can fly, and they really like going to hang out at dairy farms and poultry operations and other farms because there's other animals there, there's other birds there that they can hang out with, there's food, there's water, there's shelter, and again, they don't get very sick. And in birds, H5N1, influenza viruses in general are gastrointestinal as well as respiratory. So you're not just worried about sharing air with the bird. They do have pretty small nostrils in their beaks, but you have to worry about that bird taking a crap on you. Yeah. And when there's, birds, ducks like to poop, and they get it all over their feathers and stuff. They're not the cleanest animals. And so that's how humans end up getting infected. That's one route by which humans end up getting infected. Because if there's bird poop everywhere and they're around these birds and they don't know that they're actually sick, then you can get exposures. And that happens to other animals, and other animals eat other animals. So you've seen a lot of carnivores. We basically find them and they're dead and they test positive for H5N1. Some of them will have pathology. It seems like eating the virus, at least for some species like cats and skunks and foxes, they end up getting this brain disease. So H5N1 definitely can replicate in the brain. There's all these scary signs, and like the money is running out for the national milk testing strategy. So I think we're probably going to see state by state less testing of dairy cattle. There's a real push to say that, there's no more bird flu here. Like it's all good. But I think when the fall comes, the birds are going to start migrating again. Seasonal flu is going to pick back up. And we're going to be, once again, I think, in a very precarious situation where a bird flu pandemic could happen. And I'll stop there because I can talk all night. But, I'm sure being big listeners of Vincent's, like there are multiple mechanisms we could talk about by which H5N1 could become adapted to transmit efficiently between humans. And that's really the one thing that it needs to do to become a pandemic capable virus.
So if a dairy farm is positive for H5N1, is that milk not used, or does it get used because it's pasteurized anyway?
That's a very interesting question, because I was just talking to a journalist yesterday about how it's kind of unclear.
Yeah.
So in the past, I was invited all through 2024. I was invited to these H5N1 expert briefings that were given jointly by the USDA and by the CDC. And from the USDA, the lead for the H5N1 response, who is the Deputy Undersecretary of Marketing, Eric Diebel, who is a veterinarian, he would tell us that in some herds, basically, if one cow tests positive, then the herd is considered positive.
Hmm.
But it kind of contradicted with something else that he said, where one of my colleagues asked, like, how do you keep the milking equipment decontaminated? Do you decontaminate it between each batch of cows that you hook up? And I mean, some of these dairy farms have like thousands of cows, and they get the cows in there and they just line them up and they hook up these claws. That's what they call them, the milking machines, to their udders, and they milk them, and it takes like 10 minutes, and then they move in the next group of cows. So she asked, how do you disinfect this between cows? And they said, we can't. So we milk the infected cows last. And so she said, well, how do you do that if like you consider all of the cows to be infected? And it's like, oh, well, we can quarantine and isolate them. And how do you decide that though if you're not testing? Well, we decide based on symptoms. And obviously, when this first happened, I mean, I think it was Andrew Bowman at Ohio State who went out and like bought a bunch of milk from the grocery store, and like 40% of it was positive for H5.
Yeah.
So
By
clearly it's
PCR.
getting,
By PCR, right?
yeah, by PCR. So the milk is safe if it gets pasteurized. But if you don't know, then
Yeah.
you're going to be selling milk that is. And it's not really clear from state to state and even from farm to farm what types of quarantine measures are always being put on these operations. So I don't actually know if they've sold milk from, knowingly from potentially infected cows, just under the assumption that it would be pasteurized. Uh, but obviously, we've found plenty of RNA in
Yeah.
pasteurized retail milk, so some of it's getting through.
Melinda wants to know how much value added do you see in the new pneumonia shot for someone who had the older shot? So this is a good question because I went a couple of months ago for a checkup, and my doc said, "This is the last pneumonia shot you're ever going to get." And I said, "Why? Because I'm 72?" And he said, "No, no, because this is the new stuff that lasts a long time." So I asked
Yeah,
Daniel, and he said, "Yes, the newer pneumonia shot. I don't know the name of it. Is will last a long, long, long time. So that's the value of it, right? Because the other one
yeah,
you had to get every 10 years, I think, or something
yeah.
like that.
That's right. And why am I don't know why I'm blanking on the name right now, but Prevnar.
Prevnar,
That's
yeah,
what it
Prevnar.
is. Yeah, my mom asked me the same question actually, and conveniently, I'm as an editor at the Journal Vaccine, I have read 8 million manuscripts about pneumococcal vaccines. So, yeah, the new one is good and it's really durable, and I believe it has additional strain coverage too.
All right, Hypoautonomic says, I believe in clots, but I don't really buy microclots as a thing. All I've seen is nonspecific dye stains that stain aggregates and a lot of coagulation proteins like fibrin aggregate, especially in vitro. Okay.
I think microclotting in vitro is kind of garbage. Like, the coagulation process as it happens in COVID is something that happens as part of pathogenesis. And you can't really study pathogenesis in vitro systems. I mean, you
Mm,
can you can address some things with like organs on a chip and stuff like that, but you really need to look at processes like coagulations and deposition of microclots as something that happens in the context of the whole organism.
right.
And while I buy microclots as a thing, I mean, clearly there are coagulation products that can accumulate in places. The real question, I think, is like, does it cause disease? And does it cause long COVID? Is it related to that? Certainly in acute COVID disease, like that can have an effect, because people have had these, increased coagulation times, and they've had evidence of coagulopathy basically. But long term, is is it something that's contributing to long COVID? I mean, what is long COVID also? Long COVID is actually a collection of many different pathological conditions. So it's really kind of hard to wrap your mind around this, but I think this is one of those problems that you can only solve so much by looking at it in cell culture.
Very Nika says, as a Russian, I gotta say most of us do mind when it's minus 40 C.
Alexi does too. I don't know why he was bragging about this because he doesn't walk the dogs on days that it's minus 40, even with his fur hat on and his fur ushanka. I bought him one of those Russian hats. He still doesn't like it when it's minus 40. And I don't either. I don't know why people live in places where it's minus 40.
Mark says, glad you are in Canada, safe from our administration of idiots. Well, you have your own problems there, don't you?
Yeah, we have our own problems, and they still talk about making us the 51st state. So maybe I just went north to find myself still in the U.S.
All right. So Tryon wants to know how you can prevent accidental intravenous injection of mRNA vaccines, right? Because there's this. Do you believe that some people who get a, myocarditis is because the vaccine was accidentally put in a vessel?
I've never seen any evidence that suggests that. So, I mean, no. I'd have to see some evidence that did suggest that. As far as avoiding like accidental IV injection, though, I'm not a clinician. I could tell you how to do it on a mouse's tail, but I don't know how you could avoid that. I think the biggest, I actually don't think that would be that big of a risk, though. I
Yeah,
mean, I've
Daniel's.
never accidentally IV injected like an animal when giving
Yeah.
an intramuscular injection.
So for a while there were some people were pulling back on the needle before putting the vaccine in, just to make sure you're not in a vessel. But Daniel said that is not really necessary. So
Yeah, I mean, I guess I just don't know that much about it because I've only really given ever injections to mice, but it doesn't, we have like an 80-year history of giving vaccines by intramuscular injection, and this has never been a problem for any other vaccine, and I just don't see why it would be for these vaccines.
Christina says, I was going to ask who is funding Ebola research, but if you have security clearance, I guess that answers my question.
Not really. I've had Ebola funding from NIH, from NIAID. I've had it from DARPA, the Defense Advanced Research Projects Agency. I've had it from DTRA, the Defense Threat Reduction Agency, and I got a good score, and hopefully I'll get it from the Canadian Institutes of Health Research on a future grant application. But there's a lot of reasons to fund Ebola research. So I want to make it really clear that the reason I have a security clearance, and it's not like a top secret security clearance, right? Like I don't get to look at secret documents. This security clearance, both in the US where we call it select agent clearance and in Canada where we call it HPTA, or the Human Pathogens and Toxins Act, basically says that I can be trusted to have access to my lab and to work with these pathogens. And I'm not a risk for doing something malicious or very careless with them. It also indicates that, I've had a background check. There's nothing suspicious in my background that would suggest that I'm getting this job so that I can get access to these dangerous pathogens. And this is something that I think is really important for people to understand, because there's, I think, a misconception that when virologists are working with these dangerous pathogens, that our goal is to somehow make them more dangerous. And it's actually the opposite of that. Like, I don't work with any of the viruses that I work with, and that includes seasonal flu and stuff that is BSL-2 containment. I don't work with any of these viruses with the expectation that it would be fine if I just got them. I try to avoid getting lab-acquired infections as hard as I can, and biosafety is my first priority no matter what. The reason I do the research I do, whether it's funded by the Department of Defense or the NIH or CIHR, which is the Canadian equivalent of the NIH, is to actually improve health. And so all of the defense grants that I've had have actually focused on the national security issues that would happen if you did have a big outbreak of this. So it's it's really biodefense, but it's not just biodefense in the sense that you're defending against a possible terrorist attack. You're also defending against what the consequences would be if there were a catastrophic outbreak for natural reasons. So this is something that, it's important that the people who work with these viruses can be trusted with them, that I'm not going to just like, oh, I'll take some H5 out of the lab and, see what happens, like, and go home and have dinner with my family and not worry about this. Like, absolutely not. And like I was saying a little earlier, if I even have a sniffle, like if I don't feel 100%, I will stay home from work and I'll report it to our biosafety team if there's any possibility that it's something that I could have gotten in the lab, because that is part of our protocols. And it is really important that you are trusted, and that you are not going to take advantage of some loophole, that you can be trusted to report a biosafety failure, whether that's intentional or not. And I really want to make it clear that the security clearance here is not so that because I'm weaponizing viruses, or because I have access to weaponized viruses, or that I'm trying to do something that, would make these viruses more dangerous. The reason I work with these viruses is to try to, make people more safe from them.
Very Nico wants to know if there are sex differences in outcomes. I wonder what differences there are for pre and postmenopausal people.
Oh, that's a good question. I'm in my mid 40s, so I would like to know the answer to that myself. I need to find the right model to do it, though. Unfortunately, mice are limited in what you can do with them because they don't actually have similar menstrual cycles to humans. They have something called estrus, and so their reproductive hormonal cycles are a little bit different. So we can look at the impact of hormones overall on pathogenesis, but looking at stuff like pre and post menopause, like mice don't really go through menopause, and there are models of it, but that's probably something that you'd have to design a more complex, like maybe clinical study for, or maybe do it in non-human primates. But that sounds incredibly expensive in non-human primates.
Yeah. Costello wants to know if mice or smaller mammals carry rabies at all.
Well, bats do.
Bats, foxes, skunks, and raccoons in the U.S. are the main ones, but probably other mammals can carry it as well. I mean, Veronica says hedgehogs, dogs, of course, and there are lots of wild dogs all over the world, and they have them as well. Yeah. And the reason she asks is, of course, I live in the forest, and I'm really curious about getting a preemptive rabies shot. I don't know if they'll give it to you. Do you know, Angela, if you can just go to your doc and say, I'm have a lot of contact with wild animals. Can I have a rabies shot?
Yeah, you can, but you have to pay for it, and it's pretty expensive. I've gotten a rabies shot. I went to Indonesia in 2012, and there was a rabies outbreak in Bali where I was visiting, and so I had to get the rabies shot for that. And then years later, I have a bat-related project that I didn't mention to you. Actually, I'm also working on bats and their host responses to viruses from wild bats in Nigeria. But in order to handle any bat, and I didn't end up going to Nigeria, but I went to Puerto Rico instead with my collaborator, where he has a much safer bat field site where I wouldn't run the risk of being kidnapped and held hostage. And there's still a big risk, though, of rabies. And so I had to get my titers checked. And the problem with the rabies vaccine is it's not super durable. So I was right at like the lower limit of what your antibodies should be. So I ended up getting a fourth one. And because I've had all these work trips to get it, I've been very lucky, but that's about $1,200 worth of rabies shots, US. So if
Don
you can
wants
get it by going to like a country where rabies is endemic, it's a lot cheaper. So you could also do that, but you can get it. You just have to pay a lot for it.
So Don says, do the forest fires and smoke we are experiencing affect the spread of viruses?
Oh, and Don is from Regina, which is the capital of Saskatchewan. It's about 2 hours south of me. So we are getting a lot of smoke and forest fires. And I think the reason I stayed home from work yesterday with respiratory symptoms was actually more that I was having upper respiratory tract issues because of the smoke from these fires that are in northern Ontario. And the problem with living on the prairies here is that if there is smoke, if there are forest fires basically anywhere in northern Canada, and the wind changes direction, like it all flies right at us, and it's incredibly smoky and difficult to breathe. You know, the forest fires and smoke probably don't directly affect the spread of viruses, but they have secondary effects that could. And one of them is just if you're around in the smoke, breathing it causes respiratory distress. And that actually can make you more vulnerable to getting different respiratory viruses. And
I
actually,
also
during my graduate work, that was like the thing that if I had stayed another year in Vincent's lab, maybe I would have given mice the common cold, because disrupting their respiratory mucosa, I was convinced was like the way that I was going to give a mouse the common cold, but I didn't have enough time to passage my virus before I graduated.
so
So,
smoke and fire can affect animal reservoirs of viruses. And so I'm
mhm.
thinking of the Nipah outbreak in Malaysia, where they were clear cutting forest and burning it, and lots of smoke. And the bats who lived in the forest were forced out, and they went to the piggeries where there were mango trees planted in the outdoor piggeries to find food, because their food source was destroyed by the smoke and the fire and so forth. So you can imagine other animals moving around with their viruses and potentially spreading them to new hosts as well.
yeah, deforestation is a huge cause of this. And I don't know if Dawn likes fishing like many Canadians do, but last year we went up to a place that is about, people from Saskatchewan know, 3 hours north of L'Orange. So it's about 7 hours from Saskatchewan. And there was so much wildlife there. Like, it was really, really striking. It's this Canadian Shield Lake that's in the middle of nowhere. There's all this pristine forest around there. And I mean, the guy who owned the lodge was casually telling us like how annoyed he was that he has to shoot wolves because they come onto the property. And if they come onto the property, that means that basically they're going to try to start getting into stuff because they see it as a place where they could source food. So you have to shoot them.
Mm.
And they're not endangered up in this part of Canada. But I mean, of course, part of me was like, do you take samples from them? Like, would you mind taking a sample and putting it in your freezer for me? They did mind, apparently, sadly. So I'll have to get some grant funding to actually go up north and actually start sampling some of those animals. But deforestation is a huge cause of ecosystem disruption. And like Vincent said, having these animals driven out of their habitat and into other habitats where they're going to come into contact with other species and other people has really unpredictable effects on like virus ecology. So that definitely is another factor.
Hypoautonomic wants to know if rabies circulates long term in raccoons, or are they getting it from a reservoir species? They are the
That's also
reservoir, right?
maybe. I mean, raccoons die from rabies, but so do bats. And I don't know, actually, Vincent, what do you think? I mean, I'm sure there's some species of bats that probably don't die from rabies.
Yeah, I'm sure. I mean, people
We
say
just haven't
bats
found.
don't get sick, but I think they do and they don't. But raccoons also don't all have to get sick. I mean, that's a major reservoir of rabies virus, at least in the U.S. In other countries like India, it's wild dogs. So it must be passing through the raccoons, and some of them may die, but others may survive and pass it on to others. And it's one of the reservoirs, I believe, of the virus in nature, besides bats, right?
I'd
And
actually
also,
really like to determine the LD50 for rabies for some of these carnivores. I mean, I don't know how you could ever get funding to do something like that, or have a containment lab where people are going to have a bunch of wild carnivores in there infected with rabies. But I mean, I think it would be really interesting because we've always made this assumption that everything that gets rabies dies from it, like people do,
yeah.
and I'm not sure that that's true.
What's the containment for working on rabies virus? Do you know?
It depends on which strain you're working with, at least in the US. since I don't work with it, I haven't brushed up on the rules in Canada. But if you're working on some strains in vitro, like the strains that they use as vaccine vectors and things, then it's BSL2. If you're working with something that's pathogenic or you're working with it in an animal, then it's BSL3. I don't think anybody does experimental rabies infections, though, like dogs. I mean, I don't see why you would really need to now, because there is a vaccine that clearly prevents it very well in dogs.
Yeah.
And rabies is one of those viruses. It's like polio. It's one of those viruses that's really scary, and we haven't answered every question about it. But for a variety of reasons, it just doesn't get worked on as much.
As of June 25th, nobody can work on it unless you have a special dispensation, a
I bet
polio
that's going to
essential.
change when we start seeing polio cases in the U.S.
Oh, is that what's going to take to get people sick again, to change the ruling?
I
So
used to think it wouldn't, but kids are dying from measles now, and I feel like people are going to have to see what happens when American children get polio, which I have nightmares about.
So MP wants to know, how would a vaccine RNA even integrate into DNA? We don't have a reverse transcriptase or an integrase, do we? Actually, we do. Cells have reverse transcriptase, and genes move around in our nucleus all the time. When an mRNA is made, it's copied into DNA and it goes somewhere else and integrates. That's how transposases of certain kinds work. So there is reverse transcriptase, but it's very, very low level. And
Yeah.
the vast majority of mRNAs in your cell are not copied into DNA. So, the experiments with mRNA vaccines and reverse transcriptase were done with cells overproducing reverse transcriptase, a completely artificial situation. So I see no evidence of integration, but that's how it would happen, MP. That's the idea. All right.
Can you actually even reverse transcribe like the mRNAs with the modified uridine? I don't know.
I don't know. It's a good question. I think in some of these studies they actually did take the actual vaccine, the Moderna or the Pfizer vaccine, and put it into cells, and so I think it can be probably.
Was that line transposase preprint that
Yes,
came out?
yes.
Yeah.
They had a line overproducing cell line, and it's
Yeah.
totally. Melinda says, what I have heard is that the spike protein is causing worse cancer, not the jab. Just say that that's not true, right, Angie?
Yeah. That's not true. I don't know even how that would happen. I mean, the spike protein, viral glycoproteins can do a lot of things, but the spike protein pretty much helps the virus get in. That's
People
it.
make up stuff all the time to scare you. To scare you into getting vaccinated, basically, right?
Yeah. I mean, I just think to scare you out of getting vaccinated in this case. And I mean, so I guess I don't have that much of a worry about cancer because I've had a lot of bad sunburns in my life and I used to be a smoker.
Mm.
But my feeling about this is that I've never seen any evidence that vaccines cause cancer. The only thing I've ever seen about vaccines is that some of them actually prevent cancer. And so to date, I mean, hearing rumors on the internet or reading a paper like the one Vincent and I were just talking about, which is not a very convincing paper in terms of the reverse transcription abilities. I just, unless I see some evidence, I'm not going to live my life in fear of mystery cancer from the internet over preventing diseases that I know could kill me and that I could pass to people that I care about and people in my community. Where a vaccine, the risk benefit analysis, vaccine versus spike protein is making cancer worse. I'm always going to side with vaccine.
Elizabeth says, self-important and stupid perfectly describes everyone who signed the Great Barrington Declaration. It's just ridiculous. All right.
10 out of 10. No notes, Elizabeth. I agree.
Renzo, hello to both of you. Nice to see Angela. I have seen you interviewed here on the CBC National Canadian News.
I'm
Renzo's,
glad, I do love the National. It is the nightly Canadian news show. And what I love the most about the National is I will get a call from one of the producers or one of the reporters, and they'll have like a 45-minute conversation with me on Zoom. And then the National includes so many stories on it every night that it always ends up like being like a 10-second clip of me being like, make sure that you're updated on your vaccines, like stay healthy, everybody, like kind of thing. It's really funny, but they're really good, and it's actually really refreshing to be in Canada and watching the news because it really is like the national news.
Okay, here's one. Jennifer. Is it possible to use the number of differentially expressed genes, or DEGs, to examine the lung transcriptome of animals found at HSM to prove they were infected and shedding SARS-CoV-2?
Well, that's my dream experiment. Unfortunately, we don't have samples from the lungs of those animals found at the Huanan Seafood Market. However, I am working on this right now, and the only reason I haven't pre-printed it yet is that I know that it's going to get picked apart quite a bit. But I have actually looked at different transcripts from different species that were found in that environmental swab data, and it's never going to show like that there was an intermediate host that was infected. Like you just can't do it with environmental data. But this was reported on in December because I presented it at this conference in Japan, and there were a couple of reporters there. So both Nature and Science reported this, and I found signatures in these environmental samples that were consistent with infected humans, infected raccoon dogs, and infected greater hog badgers. So again, it's sort of slicing the salami a little bit thinner without actually making the sandwich. So it does narrow down the list of suspects that was at the Huanan Seafood Market. It confirms that humans there might have been infected, and then these two species that we also know are now susceptible because I tested susceptibility in the lab. Are also showing signatures that are consistent with infection with a virus, but it might not necessarily be SARS-2. It might not necessarily actually be anything because these are environmental samples and they're the worst samples that I've ever worked with in my entire career. But we've done as much as we could to try to do that. Unfortunately, we can never identify differentially expressed genes because differential expression is dependent on having an appropriate control. And we can't have an appropriate control with these environmental samples.
Tona says maybe it would be better if he had, this is Mark Thompson, called you Rant and Yell-O. Because I do rant and I do yell sometimes. That's right.
Well, that's great, Vincent. I mean, it's great to have me as a guest here because I'm also known for ranting and yelling and also swearing.
They didn't get it from me.
No, I as you know, like I came to your lab already having those skills.
That's right. That's right. John says they tried to make him rock and roll, but he rapped back black and yellow. That's very clever, John. Very good.
You're not from Pittsburgh, though, like Wiz Khalifa, who sings
That's
the
right.
black and yellow song.
That's the best way to remember how to pronounce my name, right? Black and yellow.
Yeah.
You just replace the R, rack and yellow.
It's pretty good.
Yeah, one of my students years ago at Columbia in the undergrad course said, Professor, that's how we remember your name.
Really?
That was very funny.
Yeah. That's great.
Christina says, I laugh at the mention of difficulty in growing virus in culture. See this often in GMP upstream processing. Baculovirus, for example, often requires special care and additional passages to get to 50 liters. I mean, scaling up to the 50, 100, hundreds of liters is very, very different. And very
difficult.
Like
I want to like cast myself into the sun, like thinking about trying to make 50-liter cultures of rhinovirus 1A.
Yeah, right. Mark wants to know, Angie, do you have dishes in Vincent's wall of polio?
No, that was after my time. And I actually didn't work with polio that much. I did a little bit of mouse work with polio, and I learned how to do a plaque assay with polio. Polio is really such a beautiful virus to work with. It's one of the worst viruses in the world. And I mean, I say that from my position now as somebody who specializes in viral pathogenesis, because it really is one of the worst pathogenesis phenotypes I've ever seen in a mouse. But the virus itself is so great to work with. It's just easy to clone. It's easy to grow. It tolerates all kinds of terrible conditions that every other virus like mostly hates. It's a really lovely virus, but I actually didn't work with it that much in Vincent's lab. Instead, I worked with horrible rhinovirus 1A, which was much more of a diva than polio was.
Tryon wants to know why rhinoviruses replicate at low temperatures. Oh, we had this discussion years ago. Not all of them. This is a myth, actually. I think they do pretty well at 37 also, right?
I grew almost everything at 37 because I was trying to get a virus that would grow in the lower respiratory tract. So I wasn't trying to maintain like just the upper respiratory tropism. I didn't have a problem with growing any of these viruses. I know back in the days of yore, before my time, Vincent, that you guys would make these temperature sensitive mutants quite a lot. But you know, the questions that I work on now and even in grad school, like what I was working on really didn't depend on growing things only at 33 degrees, which was for whatever reason the decided like temperature of choice for the rhinoviruses. But there have been some studies where people have shown that like interferon responses are different when things are grown at different temperatures. So, I mean, it may be important. I've just never really focused on that. And when I was working with rhinoviruses, I was trying to make them infect the lungs so that I could have a model that you could use to look at things like asthma pathogenesis and stuff like that. So I needed it to at least get into at least the mid respiratory tract where it's 37 in a mouse. So I just grew everything at 37 for the most part.
So Elizabeth says, I get geese and ducks in my lawn and worry about mowing around the areas they poop. A lot of people ask about this, and also, is it okay to have a bird feeder and have wild birds come? What do you think about these issues?
I think just be careful about it. Like, I mean, you know, in some places, like in Seattle, for example, you cannot avoid like goose poop in some, like the Canada geese are everywhere. Yeah. And especially if you're near like a body of water or something, like it's just completely unavoidable. I mean, people can, you should just understand like everything else, it's a risk, you know, and take precautions to minimize that risk. So you can mow the areas that they poop, for example, but, you know, maybe do it on a day where it's kind of windy or something. I've never heard about anybody actually getting flu from a lawnmower, like, or actually getting any virus from like aerosolization from a lawnmower. I suppose it could happen, but I mean, it's outdoors.
You can get tularemia from mowing over a dead rabbit that has tularemia, right?
I bet you could get H5N1 that way too. So don't mow over any dead birds. But
it's called what is it called? Lawnmower disease, I think.
Oh my God! Like,
and I was talking about it on some program, and that guy sent in a question. He says, "My lawnmower is having problems. Can you help me fix it?" No, it's not the disease of the lawnmower.
Yeah, yeah. Tularemia is something incredibly different from a broken lawnmower. But I mean, with any of this stuff, you know, like I worry about this too. My husband last year, because we have a big backyard. It's very suburban here. And we have this big fence in the backyard that like splits off our house from a berm, like that is across from a street. So it's like city property over there. And my dogs, I have 2 dogs, they were just like obsessively staying in this corner of the yard. And so Alexi, my husband, goes over there to see what's going on. And I was at work. And he's like, you know what they were doing? They were sniffing a dead bird that was back there. And I'm like, what did you do with the bird? And he's like, I threw it over the fence into the berm. And I was like, why didn't you put it in a Ziploc bag? Like, I wanted that. And he's like, you wanted a dead bird? And I'm like, wait a second. Did you go wash your hands after you handled that? Did the dogs like chew on it? How did that bird look? Like, and he's like, I don't know. It was a dead bird. And he was not very good at providing me with information about the possible influenza status of this bird. And he didn't keep a sample for me to test. Mm-hmm. However, I don't think he'll make that mistake again. But people encounter this stuff all the time. Most of the time, it doesn't result in a spillover that causes disease, but it can. So anytime that you're doing something where you could be exposed to something like this, and that includes goose and duck poop, just use precautions. If you're outside and you're mowing the lawn, it's probably not that big of a deal. Maybe it would be a bigger deal if you ran the lawnmower over dead animals that are in your yard or something.
Well,
You don't want tularemia either, by the way.
if you're concerned, wear a mask when you mow the lawn, right?
Yeah, exactly.
So
Um, that's what I would do. But I have to say, I wouldn't be that concerned. And we do have quite a lot of birds around here. I mean, granted, I'm probably more up to date on like I would be aware if I started becoming symptomatic for H5, like probably
yeah.
before most other people would. But at the same time, I mean, exposures like that are really chance encounters, and people mow their lawn all over North America all summer, and at least as far as I'm aware, nobody's gotten a serious case of avian influenza from it.
It just takes a perfect storm, Angie, of someone
Yeah.
mowing a lawn and there's a carcass on it with a high level of virus, and who
if
knows,
i
right?
if I start a pandemic by causing somebody to get H5N1 from mowing their lawn, then I promise I'm going to quit virology. And I'm going to quit talking to the press. I'm going to move somewhere like off the grid and probably die of H5 myself.
So Vista wants to know if you should worry about H5 from eating birds and cows. Can it be a gastric infection in humans?
That's a very good question that we don't know the answer to. There was one child in San Francisco who was reported to have been sick from consuming raw dairy products that were H5N1 positive, but that kid never had a conclusive H5N1 test. So we don't really know. But one thing is very clear, and that is that pasteurization of dairy products inactivates the virus completely. So it is safe to eat pasteurized milk. It's safe to eat cooked meat from chickens or cows. The problem that I would worry about, and I certainly would not drink a glass of raw milk. And actually, I'm lactose intolerant, so I probably wouldn't drink a glass of milk at all. But I wouldn't consume raw dairy products, and especially, maybe I have consumed raw aged cheese, but I would not consume raw milk for other reasons too. I don't really want to get Campylobacter or Salmonella. There's really no reason to consume unpasteurized milk. You definitely shouldn't ever be eating uncooked chicken. I don't think that you would be likely to get H5N1 from like a rare steak, because for the same reason that you won't get E. coli from eating a rare steak. You cook the outside of it, and it gets rid of anything that's on the surface. H5N1 does not appear to actually infect cow muscle tissue, and it really is. It's kind of weird in cows. It doesn't really make them very sick. It seems like it does have a pretty big effect depending on what route they're infected by. Like, so if you infect a cow by the intranasal route, it's not always going to go to their udders. You can infect them by the intramammary route very easily, but there's now some conflicting data about whether that happens in the real world. So there's still kind of a lot we don't understand about like what is getting infected in the cow. But the bottom line is, if you cook it, you're not going to get sick from it. Cats get very sick from it, and that's probably what we have the most data on in terms of flu from eating it. They develop a very severe neurological disease that doesn't really look like human flu. I wouldn't want to take the chance of getting that. So again, no raw milk for me. But other than that, like, I don't think the risk is very high that you would actually consume enough virus to potentially get sick. And we don't really know, I mean, what the route is for that. Like, maybe the cats are getting infected orally, and the fact that they're eating it is irrelevant. I actually have a grant going in to study this.
Well, when you chew something, you make an aerosol, which could theoretically get into your upper tract, right? Right. So here's a great one.
Also have the receptor in their esophagus. So it's possible that it gets there before the stomach. But I don't see how influenza virus would actually survive the stomach. It's not
Yeah,
polio.
right.
It has an envelope, so it can't really.
Here's one from John. This is perfect. Laundemic.
I love it.
It's good, right? This Angie's going to start a laundemic.
I'm gonna try not to, and at least I personally don't mow the lawn. That's my husband's job, as almost every other household chore is. So it's Alexi's lawn demic if it happens.
So Kang wants to know, how does wildlife monitoring inventory viruses found in an animal? You should know about this, right?
Sort of. This is why I don't really do virus discovery work myself, because you can't really find all the viruses in an animal. And this is one of the reasons I like being an experimental virologist for the most part, rather than somebody who's doing a lot of field work. I also don't really like camping that much. But I like being in a lab doing the experiment, because then I can monitor the infection from start to finish, like in the animal, and relate different aspects of the infection to the disease that it causes. When you get an animal in the wild, when you're doing surveillance or virus discovery, you're just taking a sample from one part of the animal, if you're not doing lethal sampling. And then, you might get that animal on one day that it's sick. You might get that animal one day that it's not. And you're just looking at that animal in a very small slice of time. It could have, really high virus titers tomorrow, and you just missed it because you went sampling today. So a lot of this is really looking for a needle in a haystack. You will find a lot of viruses in wild animals, but the vast majority of them are either viruses that you've never heard of before, viruses that don't seem to cause disease, viruses that are unclear how much of a pandemic threat they could be, which is actually where I come into this, because the next step, I think, is to try to look at some of these viruses in the lab and try to figure out which ones we actually need to worry about in terms of there being threats to humans. But you can inventory as many viruses as you can find, but that's not necessarily going to tell you a lot about what those viruses mean, other than they all are in that animal at the time that you sampled them.
So Patricia wants to know, are we losing our immunity? She's talking about SARS-CoV-2 on a population level. I can't get Novavax for some time to come. Am I alone in worrying about the deliberate decline in COVID vaccination? Yeah.
Well, yeah, I mean, no, you're not. I'm worried about the decline in all kinds of vaccinations, which I think is what's coming. I think we are losing our immunity in the sense that people are just not going to get vaccinated as much. We'll still have immunity at population level against all these viruses. Even this guy from Children's Health Defense who got measles, according to him, had been vaccinated with one dose of the MMR back in the 1970s, I think, and still provided him with enough protection, even if he did have measles, that according to him, his disease lasted like 2 days. Might not have been measles, but the many of the vaccines that we have last for years and years and years. I mean, the last time I got my measles titer checked, I was in 2019. I was going to Southeast Asia on vacation, and they recommended that I get it checked because there was an outbreak in Cambodia, I think, one of the countries that I was visiting. And my last MMR shot was when I was 11, and I'm in my mid 40s now. So 30 years went by, and I still had really, really high measles antibody titers. So we'll still have immunity, but our children won't, and younger people won't. For COVID, that will wane, and depending on the availability of COVID shots, same thing for flu, we'll still have immunity, just not as robust. So it's a pretty bad situation that we're in for.
RFK has taken away COVID vaccines from pregnant women and young people already, right?
Yep.
And the excuse is that they cause myocarditis, which doesn't happen anymore. And I don't know why Prasad didn't get that memo. There was a study just done. When you space out the mRNA vaccines, you don't get myocarditis anymore. So that's just a lie, right?
Because he doesn't make money by telling the truth. And now he
Yes.
doesn't make money and like have power. And both of those things, are a strong disincentive for people who have no morals and no patriotism and no respect for science at all. That's an easy choice for them to make. That's not a choice that I would ever make. Because I mean,
Uh,
effectively, you're killing people by encouraging them not to get vaccinated.
yeah. This is funny. MP Dr. Rasmussen, do you need undergrads? Just kidding.
Well, I do have one undergraduate working in my lab this summer, and I do take undergraduates, but they usually have to go to the University of Saskatchewan.
Zachary wants to know if you have any insights into how limited researchers are from doing BSL work depending on their pre-existing conditions, like someone with diabetes type 1 that wants to research in a BSL.
Well, so for every person who is working in a high containment lab, even the precautions that you take are going to depend on what pathogens you're working with. So somebody who's working on, say, African swine fever virus, which doesn't infect people, is going to take very different precautions from the ones that I take, because their concern is not so much that they're going to get infected, because African swine fever doesn't infect people at all, but they have to take biosecurity precautions to make sure that they're not bringing any African swine fever virus out of the lab with them. I have to think about that too. But also, because the viruses I work with are pathogenic to people, I also have to wear a Tyvek and a PAPR and all the precautions like that are really uncomfortable actually to wear for a long period of time. Some of my colleagues who work with arthropod-borne viruses that are BSL-3 don't have to wear the PAPR. They don't need and that's a powered air purifying respirator. They don't need to have that extra level of respiratory protection. So they will wear N95 masks and things like that and eye protection rather than wear the PAPR. I actually prefer to wear the PAPR because it's kind of like having a personal fan like on you the whole time. And N95s, as most people know from the pandemic, can be uncomfortable to wear for a really long period of time. So it really depends a lot on what you're working with. But we do have also medical protocols like for prevention. So before I ever started working with Mpox virus, I got the Mpox vaccine. And so if we have a vaccine for any of these viruses, we'll get that. If you have a condition that means that you are more at risk for a given pathogen that you're working with, which type 1 diabetes may or may not be, and actually type 2 diabetes may or may not be, then that's something that you work with the biosafety and like medical surveillance team to address. If you need any additional protective measures, and at BSL-3, you probably don't need additional protective measures because the protective measures we take are designed to prevent infection altogether. So you shouldn't actually be getting infected whether you have one of these pre-existing conditions or not. But that is something that you address with your biosafety team. And also, the one last thing is that we have ongoing serum collection. And so, sometimes the doctor, like, and the nurses who are doing this will ask, like, what's going on, just to address like your medical condition from year to year. So that way, if there's ever any indication that, oh, there was a lab leak in the past or something, or you might have been endangered because you were exposed to something, they can go back and look at your old blood samples and see maybe when that happened. So there is consideration for your existing health conditions, like when you're in there, but I don't think unless you had a physical disability that prevented you from doing like all the entry and exit procedures that we normally do, and we have to shower when we leave the lab and stuff like that. Unless you had something that physically prevented you from doing that, I don't see why it would prevent you from working in high containment.
So, let's see, I had one ready to go. Here we go. Visto says, quote, you threw a perfectly good dead bird away. Husband should know better.
I would have been happy with it if he just like took a swab from it or something, but he doesn't have any sterile
Well,
swabs.
you got
So
to give him an SOP, and then he'll know from now on, right?
I mean, he does have a PhD in protein chemistry. I mean, back in the day, he was a protein biochemist who would spend like days in the cold room, running chromatography columns and stuff. So he
Yeah,
can
but
be precise.
he's not into swabbing animals, though. That's the thing.
No, and now he's a data scientist who's into like generating code on AI chatbots and like doing stuff that I don't completely understand because it's very data sciencey and mathematical.
So Ariozone says, I could see Londemic as an HBO sci-fi series. Yeah,
I mean, it would
yeah.
have to be a little more exciting than the first scene in which somebody gets sick from mowing a lawn. I would hope there's other stuff besides lawn mowing in it.
Thomas wants to know, why are vaccination rates going down? You would think they would be going up since COVID. Is the current administration the reason?
Partly. I think the other thing is that anti-vaxxers are really exceptional propagandists, and they've been waiting for a pandemic like COVID for their entire lives. And they were really, really effective at spreading misinformation, disinformation about vaccines. And the administration certainly is encouraging it and now making policy that makes it harder for people to get vaccines. But this was vaccination rates were already decreasing in some places because of this. And I think a lot of it is because of the proliferation of disinformation about health.
John is reading Arrowsmith and the way farmers and politicians resist public health regarding pathogens detected in some cows. Shutting down a dairy farm feels relevant today. Oh, it's been a long time since I read Arrowsmith. I don't remember what's going on. That's the one about the researcher using phage, I think, to try and cure infections, working at the Rockefeller
I don't think
University, I
I've
think.
read this book, but maybe I should read
Yeah,
it.
it's good. It's Upton Sinclair.
Okay.
Sinclair
I've read
Lewis.
the jungle,
Sinclair
but
Lewis.
Lewis. Got it.
But yeah, you should read it. It's very good. I have a old dog-eared copy at the incubator. But yeah, it's about a guy trying to develop phage as a therapy, but it's also about all this stuff that goes on in biomedical research. It's old. It's from, I don't know, the 20s or 30s, right?
I like that stuff. I like hearing about how they used to run viruses through porcelain filters.
Sinclair Lewis. Thank you, John. Thank you very much. All right, now, so on office hours, we get into memes, and earlier it was rabies, and now it's like Londemic. I see Londemic as a spoof of a cross between Lawnmower Man and any zombie
I really liked
movie.
Lawnmower Man, actually. It was, I mean, it was such a movie of its time.
All right, that's it. We have 2 minutes left to say goodbye. I want to thank everybody for coming here tonight. We had as moderators tonight. Let's see, we had Les, we had Vanity Nutrition. I think that's all we had in the way of moderators. Thank you very much for coming and keeping the discussion civilized. By the way, folks, we have 135 of you here and only 103 likes. Could you please hit the like button? So we're trying to spread good science, truthful science, not misinformation, not disinformation, no lies. So please hit the like button, and maybe that will help us get more publicity. Our guest tonight from the University of Saskatchewan, Angie Rosenfeld. Oh my God, I'm combining two of my students.
I know. I was like, wait a second, what? Like,
I two students, Angela Rasmussen and Amy Rosenfeld. Angela Rasmussen, thank you so much for joining us. It was a lot of fun.
my pleasure. Anytime, Vincent.
And we will be back, I believe, next week. Let's see. Next week is the 30th of July. We'll be back. Office hours, 8 p.m. Eastern Time, U.S., right here on YouTube. Thanks for joining, everybody. See you next time. Good night.